Okay, good afternoon also from me, from Houston. This is the, I believe, fourth in a series of webinars that we are giving, always with case studies to specific areas, and today it's about neuroscience, of course. Just briefly the agenda. I'll talk about microRNAs, what we know currently, where we are with our knowledge, specifically also, of course, in terms of neurosciences. Some milestones there. Then in the recent studies, I will mention a couple of recent reviews that are helpful. Then on the current methods, I will focus on the two main methods for profiling and discovery, which are next-gen sequencing, of course, and microRNA microarrays. And then we will finish up with a couple of case studies. Some of them are customer publications, not all of them are. And they should give you a good overview about what's currently being done in the field and what you might be able to do. And of course, whenever you have questions, just feel free to go ahead, ask Chris or Jason while I'm talking, or of course, email me afterwards. If you're new to the microRNA field, chances are you haven't heard of us. We are rather microRNA-centric company. We are located in Houston. That's where our headquarters and our main lab is. If customers submit samples, they always end up here, and all the experiments are being performed here. We're a rather international company, so we have customers in more than 40 different countries. We're doing this as the first microRNA microarray service provider, so we're doing this since 2005. And part of why we are in this field is simply our microfluidic chip technology, which allows us to be extremely flexible with the probe content. So that allows us to keep up with a fast-paced environment as the microRNA research field is. Besides the transcriptomics area that we are focusing on, we also have applications for in the proteomics field for our chip. But the microRNA field has really evolved so quickly that has caught up most of our resources so far. So if we look at microRNAs, what we know so far, microRNA are small non-coding RNAs, anywhere between 20 to 24 nucleotides long typically. They're a little bit longer for plants, more towards 24, whereas they're a little bit shorter for animals, typically between 20 and 22. All microRNAs derive from a precursor molecule, which is very, very stable and very well defined. It's relatively easy to predict the sequences sometimes. It's or find precursor molecules based on predictions. They have a stem as well as a hairpin loop structure. And as mentioned, the low energy, the low free energy is remarkable. So typically under array conditions, for example, it does not unfold. Many microRNAs are evolutionarily conserved, and it doesn't matter whether you're looking into the plant field or the animal kingdom. It's true in both. The overall mechanism is, the basic mechanism is that they bind to complementary messenger RNA molecules and inhibit translation. So they act as negative regulators of translation. And in order to do that, they exist in a rather high copy number. Although recently there have been some studies that even low abundant microRNAs can have a significant effect. But in general, they exist in a pretty high copy number. Then what makes them interesting for the pharmaceutical or diagnostic field is, of course, the fact that their expression is developmental, disease, and tissue specific. This allows, of course, applications as biomarkers. That will be one of the first things that come to mind. Others will be, and of course, they can be used as drug targets themselves. So currently, we haven't discovered all of them yet, but currently there are more than 2,000 microRNAs known for, mature microRNAs known for human. And if you look at all the microRNA sequences that are currently known and validated, there are a total of 25,141 mature microRNAs, and this covers a total of 193 plant, animal, and virus species. All sequences are maintained by the, or are collected and maintained by the by miRBase, which is the public database. There are different versions. Start out, of course, with one, and in the meantime, we arrived at version number 19, and this update was just in August of this year. The mechanism of microRNAs as negative regulators is rather complex, because each microRNA can control multiple genes, and also the other way around. Each gene can be controlled by multiple microRNAs. Currently, it's estimated that roughly 1/3 of all human genes are regulated by microRNA expression. There are two main mechanisms that are suggested so far. One is predominantly found in the plant field. It's based on complete pairing. Basically, the microRNA, mature microRNA is an exact complementary match of the messenger RNA molecule. It binds tightly and triggers degradation. In the case of the animal model, you have imperfect pairing, so there are a couple of mismatches allowed. And the binding is not as strong. Typically, degradation is not triggered, but the binding is strong enough to inhibit translation. So the fact that in the animal model mismatches are allowed makes it, of course, even more difficult to predict the targets, but more on that later. If we look at the milestones, the first microRNA was found in C. elegans. It was called Lin4, and it basically showed that that it was encoding a small RNA. It was shown that Lin4 was encoding two small RNA molecules and not protein, which was really against the dogma back then. Then 98, for the first time, RNA-I is observed. Consequently, in 2006, the Nobel Prize was awarded for this discovery. And these two things together were basically the major challenge to the dogma at the time of biology, that basically stated that DNA gets transcribed into RNA, and RNA gets then translated into protein. So small RNA molecule that was not really translating into any protein but had a regulatory effect was really not included in this model. Then in 2000, LED7 was identified in humans and in Drosophila. That was done by Reinhardt et al. In Slack's lab. In 2001, a larger amount of small regulatory RNAs were discovered by the groups Bartel, Tuschl, and Ambros, and they gave this new class of regulatory RNAs the name microRNA, so we're based on the size. So since 2001, we can really talk about microRNAs. Then things evolved pretty quickly. In 2002 already, Drosha was discovered, which is part of the processing pathway of microRNAs. Here it's just really the rough outline mentioned. It's basically you start out with a primary microRNA, which gets then processed into the so-called precursor microRNA that I mentioned already, that has a very stable structure. And this then further gets processed by a dicer molecule into the short mature microRNA. Then for the first time in 2003, the relationship between microRNAs and the neuroscientific field was mentioned. The Hobart lab had a case where basically it was shown that microRNAs are involved in the neuronal cell fate. Example here was that the so-called ACER and ACEL neurons undergo a shared precursor state called ASE. And from this common precursor state, it really depended on the microRNA expression levels in which of the two functional distinct neurons it evolved. In 2003, the Stoffel lab first reported in vivo silencing of microRNAs with antagomers. And when it comes to methods, in 2004 was a big step because for the first time microRNA microarrays were available. Basically the messenger RNA microarrays were adapted to this new field. And this is exactly the time when LC Sciences was founded. In 2005 already, next-gen sequencing became available, probably at a very different cost than today. And it was used to, for the first time, used also for small RNA discovery and analysis. In this case, this was done using Arabidopsis. In 2006, miRBase goes online. This is back then it was hosted by the Sanger Institute. This was of course not the first version, but this is when miRBase went online. In 2008, for the first time, circulating microRNAs were detected in body fluids. And the most common source, of course, it was blood, or is still blood, then plasma, serum. But as it relates to this field, of course, also cerebrospinal fluid. And the latest development is certainly that urine can be also used for as a source for microRNAs. Then when it comes to neuroscience-specific milestones, we have to look at 2005. The Croce lab had discovered already the role of microRNAs in cancer back in 2002, but in 2005 microRNAs were then linked directly to glioblastoma. There were two big papers about this. Then in 2005 it was linked to Tourette syndrome. A year later to brain development, and in 2007 then to disorders like schizophrenia and Alzheimer's disease. In 2008, it was confirmed that microRNA expression plays a role in autism, and in the same year also it was the link between prion-based disorders and microRNA expression was confirmed. In 2009 then, traumatic brain injury as a research field, it was shown that those can be used as biomarkers, and I will actually mention a paper towards the end of the presentation that is focusing especially on this, specifically on this. And then in 2010, a paper mentioned the relationship between microRNA expression and astrogliosis. So while there has been already a lot of work being done compared to other fields that our customers work in, let's say cancer or cardiovascular research, neuroscience is still relatively at the beginning. When it comes to microRNA research, the information in the field is of course rapidly increasing as shown here. What are shown here are the total number of microRNAs, and the number that's shown here are the microRNA precursor molecules in miRBase, the number of species I mentioned already, and then the almost exponential increase in knowledge that we have in terms of sequence. And here, the x-axis, it's version 6.1, the miRBase version, all the way to the most recent version 19. And what you can see here is also the impact that next-gen sequencing had on the discovery of new microRNA sequences. Illumina released the first small RNA sample prep kit around this time here in 2008, and since then the number of sequences increased dramatically. With this, hand in hand goes the increase in publications that have microRNA focused. So these are the numbers of microRNA focused publications in PubMed. So why should you care about microRNAs in your field? I pointed out already, of course, that they're involved in pretty much every developmental stage, but here just a few more points. A large number of studies have identified microRNAs that are important for brain development and neuronal differentiation. Then they have been proven to influence brain maturation and also plasticity. Mechanisms that are known to be involved in psychiatric diseases. Then microRNAs play a role in multiple hallmark biological characteristics of glioblastoma, mainly cell proliferation, invasion, glioma stem cell behavior, and angiogenesis. And when it comes to applications, the first thing, of course, when you look at the clinical potential of microRNAs, the first thing is to use microRNA expression profiling for diagnostic purposes as biomarkers. And that's, of course, the low-hanging fruit, but they're valuable nevertheless. Biomarkers can be used. The least invasive way, of course, to obtain these profiles would be plasma, exosomes, or cerebrospinal fluid, basically any bodily fluid. And these allow, once a differential expression pattern or signature pattern is confirmed or revealed, these can be used to make very accurate and detailed clinical diagnosis. And if you go a step further from the prognosis, oftentimes or more and more cases are found where the microRNA expression level can reveal also a prediction about the prognosis of the patient. Currently commercially available is already a diagnostic kit which is made by Rosetta Genomics. It's focused on a different area, though. It's dealing with lung cancer, but it's pretty impressive. It can accurately differentiate between the 4 main types of lung cancer. Whereas before this test became available, the accuracy of the diagnosis was really not great. It was, I think, around 60% correct diagnosis. Then if you go into the next level, you can also use microRNAs, of course, in the drug discovery and as therapeutics, particularly in the cardiovascular field. The research is already a little ahead compared to other research areas. Basically, microRNAs can be used themselves as drug targets, or they can, of course, also act as therapeutic agents, basically as by giving either microRNAs directly or microRNA mimics. And yeah, then besides that, of course, in addition to mimetics and antisense oligonucleotides to modulate these microRNA functions, the latest efforts have been directed towards developing of basically development of small molecule drugs that can influence the biogenesis of microRNAs or influence their function. And when you look at the recent review papers, this is probably the most current and a very comprehensive one. It was published in May in Trends of Neurosciences. The title is Microarrays in Neuronal Function and Dysfunction. The authors are from the Scripps Research Institute, and basically it's a very comprehensive list of microRNAs and in which particular diseases or dysfunctions they are involved in the neuroscientific field. Just to mention a few items from this paper. Basically, it just shows a good overview that microRNAs play an essential part in all the aspects of the development, as well as function and plasticity. And furthermore, dysfunction in microRNA signaling contributes to neurodevelopmental and neuropsychiatric disorders. An example for the first category here would be, of course, Fragile X syndrome, Rett syndrome, and autism. And then an example for the neuropsychiatric disorders would be depression, schizophrenia, and drug addiction. And what is known so far is that in fully differentiated neurons, microRNAs are localized at the dendritic spines and postsynaptic densities. Then expression of many microRNAs is dynamically regulated during neurogenesis, as well as neuronal maturation and brain development. And microRNAs play also a big role in maintaining plasticity-related genes in translational repression until alleviated by neuronal activity. This paper contains two smaller graphs, but one basically just focusing on the biogenesis, a brief overview, but the rest really consists of tables such as this one. This is just a brief snippet to give you an impression. It basically lists the syndrome, the disorder, then the microRNAs that have been validated to play a role, and then the proposed function, and how these microRNAs specifically might contribute. And then, of course, all the references. So this is just for the neurodevelopmental disorders. For the neuropsychiatric disorders, there's also a table, but of course I didn't want to waste the time here. But basically, this is a very comprehensive overview about all the microRNAs and their function when it comes to neurosciences. Another paper which came out in 2009 already is focusing on a subset of microRNAs. Basically, it deals with the validated glioblastoma-related microRNAs and their potential functional importance. So again, you have the microRNAs listed, the expression levels, typical expression levels, the phenotype, and potential targets. And then when it comes to the brain field, there's a very recent paper also from this year which describes the emerging role of microRNAs in schizophrenia and autism in particular. So if you're interested in this field, this would be a source to look at. Again, microRNAs are listed here, the validated targets, and then the links, how it links to schizophrenia or autism, schizophrenia in red, autism in blue. And it's always really helpful to have validated targets because in this field, because it's still so new, a lot of times you will find the word target mentioned, but oftentimes the authors are talking about predicted targets. The last review paper that I want to recommend is not dealing really with a neuroscientific topic, but rather the methods that you can use when you study microRNAs. And I like it because first of all, it covers all the typical methods like qPCR, microarray, RNA-seq, and it does a good job in explaining the advantages and disadvantages of each method. Also as it relates to the sample type that you're starting out with. So it has two flowcharts that are shown here also. One is basically focusing on what type of sample you're starting out with, what type of methods you have available to extract the RNA. And then the other one, which is also very important, is basically ask you questions based what your experiment is about, and based on these findings, it suggests the appropriate method. And the reason why I'm pointing this out is because we oftentimes get approached by potential customers that want to use a particular method, but it turns out that that method might not be or is not really the ideal solution to answer the question that the experiment is posing. So this is a really helpful little tool too. If you take a look at overview, look at the different research tools that are available for the different steps in microRNA research, we start out with, of course, the identification, basically the discovery step. Genetic screening, direct cloning, and sequencing of those clones were probably the most common early methods. Then tiling microarrays, we have done a lot of those. Those were based typically on predictions that were found using computational methods, and here are a couple of prediction websites mentioned. And of course, nowadays next-gen sequencing is the ultimate tool to for new microRNA discovery. So once you've discovered your microRNAs in your species, you typically want to do profiling. So northern blot, of course, was used initially. In situ hybridization can be used. But the three most common ones are certainly real-time PCR, microarrays, as well as next-gen sequencing these days. And in the following, I will focus on the microarrays as well as next-gen sequencing. Of course, you can do profiling also with real-time PCR, but typically you're looking only at a subset of microRNAs if you're using PCR panels. So we typically use this PCR for validation of sequencing or array results. Then the next step would be target determination. Again, there are bioinformatic tools available. That's certainly an area that can still see a big improvement. As mentioned, the matching of the microRNA to the messenger RNA doesn't have to be perfect, so that makes things a little complicated. But there are also websites out there in the meantime that basically bundle a couple of different prediction software results and spit out the combined results. Then you can, of course, do gene expression arrays, even from the same sample that you used for your microarray profiling, for example. Or you can look at the proteomic level, how the expression changes. Other methods are then pull-down assays, 5' arrays analysis, or which is something that is becoming more and more popular in the plant field, it's degradome sequencing. In the plant field, that's easy to do because the microRNA and messenger RNA are perfect matches. So when it comes to pathway analysis, there are, in the meantime, the first commercially available programs out there. One of them is, for example, from Ingenuity. It differs from the tools that are listed here, miRFocus, miRPath, and MMIA, only in the sense as that those databases are kept up to date, the commercial ones, whereas these are also manually curated, but they typically they lag behind the recent developments in terms of miRBase content. For example, miRFocus, I'm not sure with which version they are. I believe they just updated to 18, but the website still says 17. And then when it comes to functional analysis, of course, luciferase assays can be done, knockout or overexpression models, of course, and antagomeres can be used for microRNA inhibition, or the other way around, you can use microRNA mimicry to increase microRNA expression levels. So the three major steps traditionally were, when you look at discovery, profiling, and then the validation of the results, where you run a microarray for the overall profiling of the sample to cover the entire microRNA content of the sample, and then validate specific results using qPCR. And then, of course, next-gen sequencing became more affordable as a method also in the microRNA field. So this is now clearly the tool the tool for discovery. We do this particularly for customers that work in rare species. Let's say recently we got samples from a grizzly bear, which wasn't in miRBase yet. And so what we do then is run a next-gen sequencing run using a sample or a pool of samples to cover a broad range of developmental stages, basically purely to discover as many different microRNA sequences as possible. And based on these results, then we create a custom microRNA microarray. And yeah, the combination of both tools using the strength of each we call SeqArray. Basically, in the discovery step, you use next-gen sequencing, but because the cost is still kind of on the high side compared to arrays, as well as potential bias issues during the library prep here for next-gen sequencing, we then perform the actual profiling using multiple biological repeats with these custom arrays. No matter which method you decide to use for your particular experiment, of course, one of the key points is to have a good sample. As always, there are different sample types, of course. In the meantime, there are extraction methods and protocols available for this whole range of sample types, starting from regular animal or human tissue to cell lines. These are probably the two most common ones. And FFPE samples, of course, are very valuable because you know the patient history typically. And then when you look at the circulating microRNAs, of course, in the bodily fluids, blood, serum, plasma, as well as urine, which I mentioned already, play a big role. For the extraction, you can use basically any of the commercially available kits. These three are kits that we have good experience with. The Norgem Biotech kit is unique in the sense that it's phenol-free. When you look at the QIAGEN kit, you have to make sure to pick exactly the miRNA-easy kit. There's also another kit that QIAGEN calls a total RNA extraction kit, which does not retain the microRNAs, and that kit is called RNA-easy. So make sure to pick the right one there. And Ambion has the Nirvana kit. Each of these kits comes with a variety of extraction protocols, which allows you to adjust it to a certain degree to your sample type. But if you deal with FFPE or serum samples, these vendors have specialized kits available. No matter which kit you're using, the key is to keep things consistent. So you want to use the same kit for all samples in your experiment. When it comes to Trizol, if you have a lot of experience with Trizol, extracting RNA using Trizol, then we would say, okay, give it a try. But in our experience, especially people that are new to the field, they really struggle getting good sample quality. What is a good sample quality? If you look at the QC criteria, in lack of a really good high throughput method, QC method, we are looking at the UV-Vis spectrum, and we look at two ratios. One is the 260 to 230, which should be above 1, and the other one will be the 260 to 280 ratio, which should be above 1.8. You can also run a bioanalyzer gel, which is shown here to the right. It doesn't really tell you much about the microRNA quality. They just show up here as this little bump. But it, of course, allows you to get an overall impression of the sample quality. So here the 18 and 28S peaks. Depending on the sample type, all these criteria might not even apply. If you're looking at blood, plasma, or urine samples, for example, then this doesn't really apply. We would then always go ahead and try one sample of the batch first and see how good they are. We end here in terms of RIN value. While we had RIN values that were all over the place, so we had good microRNA samples with a RIN of 3, and we had bad microRNA samples with a RIN of 10. But a good rule of thumb is that we would like to see the RIN value higher than 7. Then here are just a couple of pictures, different types of gels showing a good extraction and a failed extraction, either through degradation or simply because the kit didn't extract the microRNAs. You see here the 28S, 18S, and then here down here the microRNAs. Here in these cases, the green circles show, of course, the missing microRNA, small RNA pieces, fractions. And I would like to thank Norgem Biotech for providing us these images. Another important factor besides sample quality, initial sample quality, is of course the number of replicates, in particular the biological repeats in your experiment. There seems to be the preconceived notion among a few that just by using a very modern method, let's say like sequencing, next-gen sequencing, is that somehow you can reduce the number of biological repeats. Of course, that's not true. The statistics or the rules of statistics apply to any method. So we would suggest, depending on the experiment and the sample type, again, we would recommend at least a number of 3 biological repeats per group. And we would also recommend to always have the same number of samples in each group. Just as an example, if you're dealing with cell line samples or regular animal tissue, you should be able to get some initial statistics from just 3 samples each. If you're looking, on the other hand, at human patient samples, there you will find that the biological variation among the samples is, from sample to sample, is so big, based on age, gender, race, etc., that you will need a very minimum of 10 samples per group. And as mentioned already, it's important to have also a decent number of control samples. We know that it's sometimes difficult to get these, but if you're, let's say, comparing 10 disease samples versus just a single control sample, that really ruins the experiment. If we look specifically at the advantages and disadvantages of microarrays versus RNA sequencing, let's focus first on the arrays. They're of course around since a while. Initially, they had a pretty bad reputation based on the first messenger RNA arrays, I guess. But since they were introduced into the microRNA array field, they've been proven to be very reliable and robust. It's a high throughput method. You can really look at the entire microRNA content of any sample. Only limitation, of course, here is that you have to know the microRNA sequences of that particular species that you're working with. So if you have a well-characterized species such as human, mouse, or rat, this is certainly the method of choice. It's also very straightforward when it comes to data analysis. You get a pretty clear picture of which microRNA is up and down regulated, and it's much less convoluted than as compared to any sequencing data that you might receive. Of course, the cost is lower and the turnaround time is typically much faster. We have a rush service, for example, for 5 business days. Typical are 2 to 3 weeks. So if you look at the arrays in a little bit more detail, this is our default chip. It's a microfluidic chip which has basically one inlet, one outlet here. The fluidics, the liquid is being pushed through these channels. Here's an electron microscopic picture of these channels. basically the liquid comes in here, goes to the reaction chamber, and comes out. And in these reaction chambers, in every single one, we can synthesize different microRNA probes using photosynthesis. And the whole system is covered with glass. So the base is silica, and the top part is glass. So it's a closed system pretty much. So you have no dye bleaching or any of these issues. Because we are using photosynthesis and combined with microfluidics, we are very flexible when it comes to the content. So we can synthesize any type of probes on the chip, not just limited to DNA or RNA, even peptides. But for the microRNA field, it's very important because it allows us to really keep up to date with the latest sequences in miRBase database. So this here is showing you the known microRNA sequences in miRBase, starting all the way back from version 1 in 2003 to the more than 20,000 sequences known now with version 19. And you see this almost pretty much exponential increase in knowledge. If you're working, especially if you're working with arrays or qPCR, there are a couple of things to watch out for. There were some major changes when miRBase updated from version 10 to 10.1. It doesn't look like a big change here, but what happened here is that a lot of microRNAs were renamed as well as sequences corrected. So if you're using qPCR primers or arrays, make sure that they are not based on any of these older sequences. Then another major incision point here to mark is the switch from version 17 to 18, which happened November of last year. What happened here is that the nomenclature changed dramatically. If you look at any microRNA paper, you might find micron names mentioned with an asterisk that had to do with the nomenclature. So this whole asterisk nomenclature has been retired and was replaced by the -3, -5p nomenclature. If you want to get up to the latest version of the nomenclature, I can recommend the miRBase blog. If you go to mirbase.org, the public database, they have a link to their blog, and Sam, who maintains it, did an excellent job in explaining the recent changes and the ideas behind it. So again, if you're receiving data, for example, from us or from sequencing results that are based on version 18, on the latest nomenclature was version 18 and 19, and try to validate this with, let's say, qPCR primers, you have to make sure, or you have to be aware that these are probably still based on the older nomenclature. This is just an overall overview about the flow chart, the workflow here. In our lab, basically, when we receive samples, we do, of course, initially always a QC. Should there be anything wrong with the samples, we would contact you. Then we do the sample preparation, run the arrays, do the data extraction and analysis. And just as a brief example how that might look like is basically you're sending us samples, different topological repeats. We run the arrays, normalize everything together, and the first heat map that you get is typically looks like something like this, rather convoluted, but then applying just a couple of simple statistical methods like a t-test or ANOVA really reveals the information that's pretty much encoded in here. What you see here as a result is basically the difference between, in this case, control and treated samples at different p-value cutoffs, where the p-value symbolizes the statistical significance. And if you look at the data from the chip directly, you typically see something like this, and basically sample A versus sample B, or group A versus group B, and these clusters of signals that you see here are basically the probe repeats on the chip. So that shows you how tight they match together, how well the data of each probe repeat matches with the other. And I'll skip this. This is just basically covering what is included in the data analysis. The bottom line is you're getting the raw data as well as the fully analyzed data with heat maps and everything. If we move on to the sequencing side of things, this is of course has a major advantage over microarrays when it comes to unknown sequences. That's of course why it's first choice for the discovery purpose. It also has an increased dynamic range. It's a really truly digital solution, so what you're getting are absolute numbers for copies, absolute abundance versus relative abundance on microarrays. And of course you can, if you want to, you can look at more than just the microRNAs. You can look at even messenger RNA, non-coding RNA, other smaller non-coding RNA. The workflow again is pretty much the same as for the arrays: sample QC, library preparation, the actual sequencing, and then what is more involved here, the sequencing with the sequencing method, is certainly the data analysis part. And to give you some numbers of to give you an impression of what you can expect, what type of data you can expect, I put in this slide here. There are a couple of numbers I want to go over with. So the sequencing is based on Illumina machines here in-house. We have different ones. This is the one, depending on what the application is. For RNA-seq, we would use the Illumina HiSeq machine. For microRNA arrays, microRNA discovery, we use this Illumina Genome Analyzer GA2X. Read length are 35 bases, and you get typically 20 to 30 million reads, which of course depends on the sample quality. If you run only a single sample in this lane, you get of course all up to 30 million reads for this one sample. So that's something, if you want to look at low abundant microRNAs or simply want to do discovery, that's something we would recommend. But you can also barcode or multiplex samples, meaning you can run multiple samples in a single lane. Of course, the overall number of reads doesn't increase, so what is shown here is the number of reads per sample as you're increasing the number of samples per lane. And because of that decrease, we do not recommend more than 3, really the absolute maximum, 4 per lane, 4 samples per lane, 4 only if the sample quality is really good. Otherwise, you just don't get good data. That's our opinion. In general, typically this is being compared to data that you would get with a microarray. Only difference being, of course, that with a microarray you would have to know the sequences already for the probes. In general, if you're working with a well-characterized species such as human, mouse, rat, or in the meantime there are also others in miRBase that have a considerable amount of sequence information in there. If you just want to look at differential profiling, we would still recommend microarrays, simply because there you're using the sample in a more direct way. Hence, you have less option to bias the profile. The sample prep, the library prep for sequencing is still rather involved and includes several amplification steps. So the chances that you're biasing your profile are certainly there, and there's still discussion about that. So for discovery, we would always recommend, of course, sequencing. For profiling, still arrays. Then when you look at the data analysis part, this is just a rough overview what our proprietary software does. Basically, you get the raw reads from the sequencer. The first step is you map it to the known genome, and then this flowchart lays out how the software then determines and groups the sequence information into different categories. All the way here from group number 1, which are the known microRNA species that can be mapped to the basically sequences that were found in your sample and can be mapped to existing microRNAs existing in miRBase, and then all the way over here to group 4, which include potentially novel microRNAs, which of course is here the goal. Basically, these are sequences that were found in your sample and can be mapped to the genome, your species genome, but are not in miRBase yet. To illustrate this further, here just an example of a typical run. You get 30 million raw reads, out of which 65% could be mapped in this case. Then another 55% were out of these could be mapped to miRBase or microRNA candidates. And then here, this is the final distribution after filtering out junk and applying certain sequence filters. Basically, group number 1, which are microRNAs that were already known and were already in miRBase, we ended up with 8 million here. And then the most interesting other group here, group number 4, which are potentially newly discovered microRNAs, we got almost 30% of the reads, so more than 3.3 million. Besides the actual sequence, you get, of course, also information about the distribution of the mappable reads in terms of length distribution that might be of interest when you look at developmental stages, because that tends to shift. And then if you look at the actual sequences, you will oftentimes find that there are so-called isomers. What these are, basically, you have the main sequence with the highest count number, in this case 60,000 roughly. That would be typically the sequence that should be in miRBase. And then, but besides that, you see other sequences here. For example, the second one here, it's truncated at the right end here. And it's still considerable number of reads here, 10,000. And even higher numbers for single nucleotide mutated sequences down here. And then you have a whole variety of shorter, shifted, etc. Sequences down here. And there's still a lot of discussion about how much of this is just library artifacts and how much of this is real. Some of these are certainly artifacts of the library, but 10,000 here is a pretty high number. So that so far we think that this is certainly real. And if you look at these data, it also easily explains why you sometimes have a difference between array data and qPCR data, simply because an array cannot distinguish these top 2 sequences, whereas, for example, a Tagman array, which is highly specific, will distinguish between these two. If you look at applications, and now I'm moving on to the examples of publications. I will look also at different ones, starting all the way from basic research to drug therapeutics. And the first paper here I want to highlight is one dealing with human brain cells. Primary human brain cells and the microRNA expression levels as regulated by NF-kappa beta. So basically what was done here is control versus stressed primary human brain cells were compared, and so this was done using microarrays in this case, and then confirmed just with blood profiles. And 2 microRNAs in particular were found to be highly differentiated, and these 2 are the top 2 here in the seed map. miR146A as well as 125B. So this was published in 2011 by Lukiv's group, and this is basically a typical example for differential profile discovery between samples. Then if you look at something similar, basically differential profiling, again, which can be used as biomarker, but you switch the sample types from regular cells or tissue to cerebral spinal fluid. Then this paper is a very interesting one. It's basically, the title is Identification of microRNAs in the cerebrospinal fluid as a marker for primary diffuse large B-cell lymphoma of the central nervous system. So that was published in Blood. And basically what the authors were able to do is using microRNA expression profiling, they discovered 3 microRNAs in particular, as listed here, 19B, 92A, as well as miR21. And following this flowchart here, they were able to use the expression levels of these 3 particular microRNAs to increase the accuracy of the diagnosis to 95–96%. So that is really remarkable. As I mentioned, this was focusing on primary central nervous system lymphoma, and yeah, this is certainly the least invasive way to obtain a sample and to do an accurate diagnosis. If you look at other bodily fluids as sample types and stay in the biomarker field, this is a sample that was done using plasma samples. It looked at microRNA expression levels and whether they can be used as molecular biomarkers for traumatic brain injuries. And this was done again with microarrays, and it was very remarkable to see that you can actually really use plasma, and this was one of the earliest studies dealing with plasma, especially in this research field, that you can actually use plasma samples to diagnose a patient and to diagnose traumatic brain injury. So the analysis showed, comparison showed that out of the 108 microRNAs that were identified in healthy volunteers, almost half of them, 52, were altered after traumatic brain injury, and of which 32 decreased and 19 increased. So that was a very promising study when it comes to microRNAs as biomarkers in traumatic brain injury. This is a study that was that took it a little further. It's basically from the purely diagnostic field, was trying to venture into the prognostic area. This was done using qPCR and samples from CSF, cerebrospinal fluid. And basically what was shown here is that you can also make a fairly accurate prognosis about the treatment response in brain cancer, depending on the microRNA expression levels that you find. So this study focused on 3 main microRNAs, 10B, 200, and miR21 again. miR21 shows up in a lot of different diseases and a lot of cases. And basically, depending on the profile, you could say that, okay, this is a low-grade primary brain tumor, or on the other hand, if another profile was found, that you're dealing with a really metastatic CNS cancer. And metastatic meaning, for example, into the lung and breast. And yeah, what are shown here are different expression levels of microRNAs and how they relate to the prognosis. So this was a very recent paper just published in 2012 and in Neuro-Oncology. Then when you look at microRNAs as potential drug targets, this study is interesting. It was, again, published by Lukev et al. It looked at the expression level of microRNA 146A. The title was an NF-kappa beta sensitive microRNA 146 mediated inflammatory circuit in Alzheimer's disease and in stressed human brain cells. Basically, it was shown, of course, using, again, in this case, microarrays, that 146A played a major role in Alzheimer's disease and in this regulation, and that 146A can be used as a potential drug target. And what is shown here on the right are just a chip image from the microarray. This should be in color because in black and white it's really not that meaningful. But here the spot plot again shows clearly the increased control versus Alzheimer's disease of 146A. And the last paper I want to mention here today is one that is focusing on ALS, amyotrophic lateral sclerosis, or ALS. And this paper focuses on the effects of microRNA 206 and how it delays ALS progression and how it can promote regeneration of neuromuscular synapses. This particular picture is not actually from the paper, but it was published in Science in 2009, and in the editorial this paper is mentioned, and that's where we got this graphic from. It really summarizes the paper nicely. So basically the study shows that 206 was involved in promoting regeneration of neuromuscular synapses in mice. And here you have at the bottom, I'm not sure whether you can see that, but basically here's the type of intervention mentioned, the effect it had, and the overall outcome. So the bottom line was that 206, as mentioned, delays the progression and promotes regeneration, and can be potentially used as a therapeutic, either in its direct form or if by using a mimetic. This is a brief overview of to mention a couple of applications. Chris and Jason will be both be present at the Neuroscience meeting in New Orleans next week, starting on Sunday actually, and we will be there at booth 1917. They will be also presenting a poster there at the booth, which highlights another couple of publications. So if you want to meet us in person or want to discuss your project there, feel free to stop by. If you have any other questions, you can always email us. We have also a chat button on our website, so you can get in touch with one of us. Yeah, or stop by at the booth. I hope this was somewhat helpful to you as an introduction into the field and to give you an overview what you can do. As mentioned, our main office and all three of us are located here in Houston. All samples, no matter from where they are being shipped, they end up here. And yeah, if you have any questions, feel free to use the chat box right now, or email us at any time at service@lcsciences.com.